Hi guys, today we're going to go over nucleic acids and the monomers and polymers of nucleic acids. And I'd also like to share before we start that these are notes from Mr. David Nufke, who is an AP biology teacher extraordinaire. And he has made a whole series of notes for the entire AP biology course, which are just fabulous. So I'm just piggybacking off of his work. So we've already covered carbohydrates, lipids, and proteins. Now we're moving on to nucleic acids. So just a little bit about these. These molecules are not for structure and they are not for nutrition, typically. They're for information storage and also for enzymes. So there are two main kinds, the DNA and RNA, short for deoxyribonucleic acid and ribonucleic acid. They are made of the most common most common atoms, CHON, and also phosphorus. So if you get a question on the AP exam that tells you there's nitrogen located within a molecule, you should know automatically that it's going to be a nucleic acid or a protein. Proteins have that amine group. But if you see a phosphorus, you should know that it's going to be a nucleic acid or a phospholipid. If you see both, then you know it has to be a nucleic acid. The monomer of the nucleic acid is called a nucleotide. Joined together, you have a polynucleotide, and you could also have di or trinucleotides. But the nucleotide itself, here it is in the picture, it's made of one phosphate group, a pentose sugar, which means it has 5 sides, and then a nitrogenous base. There are 4 different bases within the DNA or the RNA, although the DNA uses thymine and the RNA uses uracil. Chained together, this is what one side of a DNA molecule would look like, or a single-stranded RNA molecule. You do need to know the main differences between DNA and RNA. And again, these are the basic ones that you need to know. It gets a little bit more complicated than this because there's always an exception for the rule in biology. But basically, the main difference, number one, is which sugar is used. They're both pentose sugars because they're shaped like a pentagon, but DNA uses deoxyribose, RNA uses ribose. And if you look down here, the only difference is right here off of carbon number 2. There's a hydroxyl group on the ribose, and it's only a hydrogen on the deoxyribose. Just one oxygen atom changes this whole sugar. The bases, I said this a minute ago, so they both use adenine, guanine, and cytosine. The DNA uses thymine, whereas RNA uses uracil. And the third difference, in most of the cases, DNA has 2 strands, RNA has only 1 strand. There are a few rare exceptions with viruses where DNA has 1 strand and RNA has 2, but those are extremely rare and they won't be covered on your exam. So DNA, 2 functions. So it stores the information on how your body is going to make RNAs and also proteins. And this is heritable. If it were not heritable, then life as we know it just wouldn't work. There would be no instructions passed on to the next generation on how to make these proteins and how to make them better over time. Okay, so the DNA structure, so it's technically, it is shaped like a double helix, and technically that double helix is 2 separate polynucleotide molecules. So on each rung, or each side of the ladder, is a chain of covalently bonded nucleotides, and they're bonded using a phosphodiester bond. That's just extra information. But nucleotides are assembled using dehydration synthesis, and they're disassembled using hydrolysis, the same as all the other macromolecules. Now, the 2 sides of that twisted ladder are attached via hydrogen bonds between the nitrogen bases. There are base pairing rules. A always bonds with T, G always bonds with C. The way I remember it is C and G are both round letters. It's just G has that little chukka on the end. And then A and T spells at, that's a word, so that goes together. Now, the nucleotides themselves are grouped into two different categories, purine and pyrimidine. So A and G are purines, T and C are pyrimidines, and I don't really have a great mnemonic for this, but I just think about like if you're taking an ag class, you're gonna be working outside in the earth and with your hands, and it's pure and back to nature. So that's how I remember purine. And then the other two just fall by default into the other category. There is a little bit of a difference in their structure. So the purines, they have 2 fused rings, and the pyrimidines have only 1 ring. That's the difference. So it's always going to be a purine bonding with a pyrimidine. And you'll see also that A and T make 2 hydrogen bonds, whereas C and G make 3 hydrogen bonds. And that could potentially be asked of you on the exam. Very brief background on this. This was an incredibly important biological discovery, won a Nobel Prize. The Rosalind Franklin failed to win the Nobel Prize because she had passed away before the Nobel Prizes were awarded, and they do not award them after you're dead. But Watson and Crick were the ones who published the paper that described their discovery of the DNA structure. It's a little bit more complicated than that. Rosalind Franklin and a partner in her lab, Wilkins, they actually did the work, the X-ray diffraction, and they had the data. Wilkins took that data and shared it with Watson, somewhat unethically. There's a lot of debate and question. If you read both sides, no one can quite agree on what happened. But at that time, England actually partitioned up research projects, and so it was Franklin's lab project to be working on DNA, and Watson and Crick were actually told not to work on it. They had other chemical problems to work on, but they did not. So there's a bit of controversy there, and Watson has been in the news. His name has been stripped from certain prestigious organizations because, well, he's a little bit racist, to put it mildly. So his work has kind of had a pall cast over it. But he and Crick did do good work using the data that was there. They discovered the double helix structure with the nucleotides on the inside, the fact that there were 2 polynucleotides, the hydrogen bonding. They figured out exactly how far apart each rung is and how far apart each twist is. So Watson, Crick, and Wilkins were awarded the Nobel Prize. Franklin passed away due to cancer that was caused by the X-rays that she used as part of her work. She X-rayed the DNA molecule in order to be able to see its structure. And she was actually the premier DNA sample preparer in the world. If you've ever done that lab in middle school where you extract DNA from a strawberry onto a Q-tip and you see that white cloudy stuff, she was actually really, really good at that extraction and getting the DNA to crystallize, which is a regular repeating pattern. She built her own X-ray machines to use the X-ray diffraction in order to get these patterns. And she was actually very, very close to the discovery of the DNA structure before Watson and Crick published their paper. If you're interested in more information about this topic, Watson actually wrote a book about it called The Double Helix. And then Franklin has passed on, but she's had 2 biographies written about her that also give information based on her like interviews and journals and diaries and things after the fact. Now, RNA is much, much more complicated. It has more functions than DNA does. As of the last time I looked, I think there was close to 42 or 44 different kinds of known RNA. These notes are a little bit outdated. I know it says 15 types. All you're going to need to know about is the three main types. We are still discovering so much about RNA and all of its many fascinating purposes. It can actually serve as an immune system within a single cell and get rid of viral DNA. So it's pretty fantastic what it does. But one of its main purposes, the one that we understand the best, is that it translates the DNA information and turns that into a protein. Um, it also can serve as an enzyme, and it serves as a regulatory aid to the cell. So it's involved in gene expression. Yeah, it is just, it's really, really interesting. And there's some pictures of it down below. But the three main types that you're going to learn about will be the messenger RNA, which is what gets transcribed directly from the DNA. Well, they all get transcribed directly from the DNA. So let me walk that back. The messenger RNA gets the message for making a protein. Okay. There's also transfer RNA, which its job is to bind to a particular amino acid and then transfer that amino acid to the ribosome. And then the other main kind is ribosomal RNA, which is combined with a polypeptide chain, and that's what makes up the ribosome. And the ribosome is actually just a giant enzyme, basically, for building proteins. Now, in contrast to DNA, the RNA is very not stable. It's ephemeral, it's made, and it breaks apart quite easily because it is exposed to so many chemical reactions. Um, Typically, it's only a single strand, but it can base pair with itself. So we see that in the case of tRNA, which is here. We call it a cloverleaf shape, even though it doesn't really look like a cloverleaf there. And there are some cases of some rare viruses having double-stranded RNA where the base pairing rules still apply, just with another strand of RNA. Now, biological systems work a lot like computer systems in a way. They store and transmit energy, or excuse me, information. They do store and transmit energy as well, but information processing basically. So the information is stored in the DNA, much like it may be stored in a hard drive on a computer. That DNA information needs to move to RNA before that information eventually becomes a protein. So it's much like your hard drive and your working memory and how that might interact with, say, your GUI to show the video on your screen. This is called central dogma. We'll have a whole unit about this. It is incredibly important to how life functions, and it's also an integral part of this course. Okay, and here's kind of an overview of it. So the DNA can copy itself and make more DNA. DNA can make RNA. Some RNAs can copy itself and come back into DNA in the case of retroviruses. And then this RNA can then go and make a protein. And here's kind of an overview of it. This is a eukaryotic cell. So we've got DNA here in the nucleus. It'll make an mRNA, go to the ribosome and join up with the tRNA and the mRNA, and then it'll make a polypeptide chain. Again, we're going to have a whole unit on that. So don't worry if you don't understand all the steps of that yet. Okay, and that is the end of the lecture for nucleic acids.